Yeah, so welcome to this webinar about the METVES II project and especially the interlaboratory comparison study. And for those who don't know me, yeah, I'm Britta and I'm a PhD student at the Amsterdam UMC under the supervision of Edwin and Rink. So I think you already saw a couple of mails from my side coming in, but yeah. So welcome everyone, and today the main focus will be on introducing the project first of all, but also introducing the protocol that I sent around to all of you. So the aim of the METVES II project is to obtain reproducible measurements of the EV concentration simply using different flow cytometers. And we're mostly interested in that because later on we want to also we want to pave the way for a reliable multi-center studies. Let me check if I can get my laser pointer. Yeah, perfect. So in the approach within the project will be to develop reference materials to calibrate the flow cytometer, and then also to develop a ready-to-use stable biological test sample that contains the VS, and that was actually my part of the project. And then both together will be used in this interlaboratory comparison study, and I'm together with Edwin and Rink responsible for organizing this study. And the idea is that we want to achieve reproducible measurements of the EV concentration using the different flow cytometers. As I said, like the reference materials should be used to calibrate the flow cytometer, and especially the flow rate by using counting beads that we developed, and they were solid silica particles. The fluorescence will be calibrated by BD custom-made MESF beads, and then the scatter will be calibrated by polystyrene beads, and then additionally we'll give a try with the hollow organosilica beads that were developed in our project as well. And just for those that don't know that, the calibration has the purpose of relating the arbitrary units that you're measuring on your instruments to standard units, such as, for example, molecules of equivalent soluble fluorochrome, or the scatter is, for example, related to a size in nanometers. And as I already said, we also developed a plasma EV test sample, or how we like to call it, a PEVTES, and that sample is flow cytometry compatible. It is pre-labeled, so it contains vesicles that are erythrocyte derived and also platelet derived, and that's because the starting material that we use for this sample is plasma. And what we were also interested in is to have a stable sample if it comes to the concentration of vesicles. And here I'm just showing very quickly, that's not too important to all of you, but just that you know the concentration of erythrocyte-derived vesicles over storage time. So I stored this sample that I prepared for up to a year at -80 degrees. And what you can see, if you're looking at the erythrocyte-derived vesicles measured on our flow cytometer at our lab, you can see that the concentration decreases by below 10%. So that's what we consider a stable sample. And on top of that, it's ready to use. So you can just take it out of the -80 freezer, and because it's pre-labeled, you just dilute it and you can start measuring right away, basically. Coming to the interlaboratory comparison study that all of you participated in, the idea is that we'll validate the reference materials that we produce within our project, and to calibrate, as I already said, all the aspects of a flow cytometer. And then on top of that, to measure comparable EV concentrations in the prepared test sample. And as you hopefully know, the study started after a lot of delays due to COVID now this month, basically. And here I'm just showing a little picture of what it looked like when the packages arrived on Monday that I was packing all the samples into. I had about 70 samples or 70 package boxes that are delivered to you and should arrive either today, tomorrow, or on Friday, depending on where in the world you're located at. The participation terms that we set, so I think all of you saw this email that you can see an image on the right. The terms to participate were that we asked people from that participated either in the EV flow cytometry work group, in the previous ISDH workshop, are METVES II stakeholders, or are part of the reference material task force of ICEF. And then on top of that, we asked that only people, or invited only people to participate that also have a track record in EV flow cytometry detection, and that's because we wanted to keep the level of the research as high quality as possible. Another thing we asked you was the sensitivities basically of your flow cytometers that you want to participate with, and we asked you especially to have a flow cytometer that's, or multiple flow cytometers depending on the lab that you're working at, that's able to differentiate 100 nanometer polystyrene beads from the background noise by light scattering to detect APC, PEVTES, and PE fluorescence, and why that is important I'm going to explain in a second. And then we also ask you that you'd be able to measure EVs directly in diluted blood plasma at a very high throughput. In total, I have about 23 labs that are participating, and we have about 35 flow cytometers included in the study until now. And all your labs are located, or the hospitals you're working at are located worldwide, as you can see on this map here, which I think is a very nice achievement that so many people were willing to participate in this study. Coming to the protocol, and that's also one of the main reasons why we're here actually. The protocol has 3 different parts, part A, B, and C, and I'm going through to explain all the different parts and what's important in them. So part A is about the requirements, first of all. So we ask you, how do you have a -80-degree freezer for the plasma EV test sample, and then also a 4-degree refrigerator for the beads and all the other buffers that we're sending around. And once you receive the packages, so you'll receive 2 packages. One has coolant packs in it, and then that are basically all the calibration beads that we provide and the quality control samples, including the buffers. They need to be stored at 4 degrees, and I'm going to show a picture of how these boxes look like in a second. And then the only sample that needs to be stored at -80 degrees, and that's pretty important here, is the PEVTES sample. And what you can see here on the right is a pretty small box actually. It's hard to see how small it actually is because you don't have the conversion, but I think it is 7 × 7 × 5. So it's a pretty small box, and this will be shipped on dry ice. So you should recognize it right away. So the protocol part A also contains a sample list, and the samples are divided into 3 different categories. Category A are the control samples. Category B are the calibration samples, and category C are the test samples. And this list also contains a lot of additional information, such as, for example, the size of the beads or the samples that we are shipping around, the fluorochromes that are attached to them, so that the concentration that the stock dilutions have. And here you can actually see, as I already said previously, what the box will look like that you should store at 4 degrees. So you'll find buffers in them. And you also find a lot of other control samples and calibration samples, and also one type of test sample, which are liposomes. But yeah, so here is basically a list of all the samples that need to be stored at 4 degrees. As I already said, they contain buffer, which is DPBS and milliQ, because different types of samples need to have different diluents. So, but I wrote that down in the protocol specifically as well, so please pay attention to that. I have VCal Nano Rainbow beads, which were an in-kind contribution from Celarcus. I have Rosetta calibration beads from Exometry, and then I have the BD custom-made MESF beads, I have the counting beads, holoorganosilica beads, and the liposomes. Please store at 4 degrees. As I already said, just repeating it one more time, please store the PEVTES sample at -80 degrees until you're using it. The protocol part A also has another part in it, which is the flow cytometer configuration. We ask you to use a sample flow that's as low as possible, but at the same time stable. So yeah, please make sure that that's the case, so we have a stable measurement and don't see some kind of peaks and like decreasing in time or something. The measurement time should be about 120 seconds, and the trigger threshold should be put on the most sensitive scatter detector that your instrument has. Additionally, the detectors that you need, as already previously asked in the email, are APC and PE for the PEVTES sample, because the PEVTES sample is labeled with or contains platelet-derived vesicles, which are labeled with an APC fluorescent marker, and then erythrocyte-derived vesicles, which are red blood cell vesicles that are PE labeled. And then also the fluorescent calibrators have matching fluorochromes, and for the Rosetta calibration beads, we have a PEVTES labeled population in there. Other than that, we ask you to so the triggering threshold should be put, as I already said, on the most sensitive light scatter detector. Please do not use an OR triggering strategy. And if you're really unsure about the sensitivity of your instrument, please select the scatter detector where you have a dim bead that has the largest separation from the background noise. And in case that you're really not sure about the sensitivity of your instrument, please contact us at study@metves.eu. And if you have any other questions, always contact us there or contact me right away. My mail address should be known to you as well. Coming to protocol part B, that's a pre-run. And the idea behind this pre-run is that we want to check the sensitivity of your instrument as well. So we ask you to measure milliQ and DPBS and the Rosetta calibration beads. And we also ask you to use milliQ and DPBS that you're usually using at your laboratory yourself, and we'll compare it to the ones that we provided. And that's because we saw with some of the instruments, also at our hospital here, that the milliQ and DPBS that we're using for one of our instruments is clean enough for this one instrument, but it may not be for some others and result in a high background count. And that's preferably what we don't want. And we know that some of you may filter or introduce an additional filtration step to your milliQ or DPBS. So we want to compare that, and then please send the FCS files of these measurements to the mail address provided. You'll also find all this in the protocol. And one person that's really important to me here is Kim, that I just want to shortly introduce, and she's a data manager at Exometry, and she'll basically be the one person responding to your emails there, doing all the analysis also for the later measurements. And within, I think we set 1 week, she'll respond and give you the go if you can continue with the entire measurement after the pre-run looks good, or additional information, or if she needs additional information, she'll contact you as well. Protocol part C is all about the preparation of the samples and then also the run of the samples. And I have basically 3 different tables in there which tell you something about the type of sample that you need to get out either of the fridge or the freezer, how to prepare the dilutions, and please be aware that the different samples use different buffers, but it's also all stated there. And we also have example calculations in there. But if for one or another reason the dilution that we are suggesting results in too little events, please increase the measurement time or decrease the dilution factor. If it comes to running the samples, please take into account obviously the suggestions that we made to set up or configure your flow cytometer. Measure according to the measurement list that can be found in table 6 here. And that's because we want to have a certain order in which all the samples are measured. You can either measure manually or using an auto sampler. That's up to you. And then we ask you to use a certain file name for our own organizational purposes, which contains the number of the measurement. So first measurement would be 01, for example, the identifier of the sample that you're measuring. This all can be found in the tables as well. The dilution factor that you used, and that's because we're using an automated software to analyze the data later on, and for that we need the dilution factor, and then the name of the sample as well. And once you have done the measurements, please send that FCS files to the mail address. You can also send it via something like VTransfer, for example, but that's up to you. There are some notes that I put in the protocol which are about, for example, measuring the DPBS, or why we're doing that, or why we ask you to do that, is because we want to check for carryover. So between some of the measurements, we ask you to simply measure DPBS in between. keep the detector settings the same for the entire experiment, please. And then also measure the noise of each detector by triggering on the noise of the PEVTES detector. And what we also want to ask you additionally is that you provide some information to us as what's the most sensitive scatter detector that you use in, what's the illumination wavelengths of the most sensitive detector as well, the sheet fluid you're using, and the flow rate that you're using as well. But all of this, as I said, can be found in the protocol as well. In protocol part D are some example plots, or like I'm just showing 3 here, but you can find example plots of all the beads and all the samples basically. So this would be just an example for the nano rainbow beads. The one in the middle is an example for the Rosetta mix. And then we have here one of the plasma EV test sample. And what we'll also ask you to do is to run a dilution series on this plasma EV test sample so that we can determine at what point your instrument hits swarm detection, because that may differ depending on the sensitivity of your instrument. Coming to the summary already, basically, the aim of this study is to measure comparable EV concentrations in the EV test sample and also to calibrate the instruments. I think on the long run also to not only measure this and bring this out as a manuscript, but also to really educate people about the importance of calibration as such and how important it is in also having or going forward to reliable multi-center studies. And with that, I'd like to thank all the people, of course, that are participants or like participating, first of all, in the study, but also, of course, the METVES II team. And then a big thanks to John Nolan and Solarcos, who in kind provided the nano rainbow beads to us, in the same to BD, and especially Tina and Majid from BD, who provided the MESF beads and made them for us. And with that, I'd like to thank you for participating here, and I hope that you have as much fun as I have in this picture in participating. And I want to also give a note about something I learned this week, which is that if you have so many samples, or in general, if you're organizing such a huge study, it feels a bit like herding cats, first of all, and you need to be pretty organized and try to keep the overview. But also, if you have so many samples, and especially a lot of them containing dry ice, or the packages containing dry ice, better not store them in a small room that does not have ventilation, because otherwise the fire brigade comes and you run into a huge trouble. And with that, I'd like to thank you for being here and for participating, and like to start the Q&A. So if someone has questions.
Thank you. That was a really nice explanation of what we all have to do. I am curious about the choice of polystyrene. Classically, we're using silica in our labs as the nanosphere. Is there a reason for the polystyrene?
I think Edwin is the best person to answer this.
I thought he might be.
Yeah. So there is of course a reason to do that. So we want to calibrate. So if you want to use the scatter of a bead and relate this scatter to the size of a vesicle, polystyrene obviously is a very wrong choice, right? Because the refractive index of polystyrene is much higher than the refractive index of an EV, and that goes all wrong. But that's not what we are aiming for here. Now we are aiming for that strategy also in this study. So we will set size gates based on, or scatter gates basically, based on the light scattering signals from hollow organosilica beads. And we have shown before that their optical properties are similar to similar sized EVs. So that's one approach, and that's why the hollow organosilica beads, the HOPs, they are abbreviated in the study, are in there. Metrologically, however, that's not a real calibration, because a real calibration relates the arbitrary units of measurement to SI units. And the only way to do that for scatter is via me theory, because the scatter to diameter relationship is so complicated. And so we need reference beads with a known size and a known refractive index and a known shape. Now, beads are generally spherical, that's no problem. Sizing is also not difficult, but determining the refractive index, that's more challenging. And of silica beads, we have experimental evidence that those beads are pure for polystyrene beads, those beads are almost pure polystyrene, so we can assume the bulk refractive index, and that we can readily measure. For silica beads, the refractive index of silica beads depends strongly on the manufacturing process. We are still developing tools to measure the refractive index in a metrological way, but they are not finished yet. So that's why we chose polystyrene beads. In the end, it doesn't matter because both polystyrene and silica beads have a different refractive index than EVs.
Excellent, thank you. Can I also ask about the choice of using EVs extracted from plasma? I think that's a really interesting choice.
Sure. I can answer that this time. So we were thinking about like the most commonly used biofluids first of all, and we wanted to have something that's resembling biofluids or vesicles in biofluids. So we actually made one from derived from human plasma and one from human urine. Unfortunately, the one from human urine didn't make it to the study because we had some reproducibility issues there. But yeah, just having like a polydispersed sample as well was very important to us.
Excellent. I think it is a good choice. It's certainly going to be interesting. I might have missed this bit, but how are the EVs labelled differently from like the other particles that will be in there?
They're labelled with CD61 IPC, which is a platelet like marker for platelets, and you can label platelet-derived vesicles with it. And then also CD23-5A, which is to label erythrocytes, so red blood cell vesicles. And I think these are the most common types to look at, like are the most common, or like most commonly studied particles within plasma as well. And especially if it comes to vesicles derived from cells in plasma. So that's why we choose those two.
Excellent, thank you.
You're welcome. So
just to emphasize that just a fraction of all the particles in those test samples labels positively. Few percent maybe, but that depends of course on your detection range.
And so just another question. So the concentration you said was about 3 times 10 to the 6 particles per mil. Is that perspective of the labeled EVs or the total particles?
Labeled EVs.
Okay.
Labeled EVs measured with our flow cytometer.
Yeah, measured with an FJ. Yeah, exactly.
So then if you have a more sensitive flow cytometer, it could be of course more.
Yeah, but that's also why we have the dilution series in there to determine like swarm detection, but also looking at the different sensitivities. So you may have a higher concentration, but I put in the sample table what volumes we have, what dilution buffers you need, and also what you can expect concentration-wise from the stock. But that's all determined by our flow cytometer.
Thank you.
Other questions, remarks, maybe as well?
It should be pretty clear then.
Yeah, or people are just tired. Could be as well.
Oh, it's getting late.
Yeah, for us it's late, but not for everyone else.
All right. Yeah, I think if there are no questions, then we can leave it to this. I think that's no problem. You all have the protocol. The protocol contains all the details. But I just gave an overview, and which was more general. So I can imagine there are not many more questions at this moment. But just email us if you have any questions.
Yeah, exactly. Just ask. Sorry, wait.
I guess just a quick one. Do you have sort of a timescale that you want this to kind of us to start sending you data and things like that? It's probably going to be after ICEV, but
yeah, I think what we set in the mail was August. But if you run into any issues, like time-wise there as well, because I heard already back from some labs that because they're going on lab conferences, that it might be a bit later, please reach out to us so we can discuss this in more detail.
Okay, great. Thank you.
You're welcome.
Actually, I think many of you will also be attending ISEF, so if you bump into Edwin or Britta, I'm sure you can also ask questions there.
Yeah, no, sure.
ISEF and CITO, so you have
And
two chances.
CITO. Oh, Jesus.
All right. Thank you very much for your attention. We are closing this call now in the recording. The recording will be broadcasted via YouTube for some people who might have missed it, just for your knowledge. And thank you very much already for attending.
Yeah, thanks a lot. Also
Thanks,
for
everyone.
being willing to participate here, like in general.
Bye-bye.
Okay.
Bye.
Bye, everyone.